Welcome back, friends. In this lecture, we shall going to see about the vesicles, and all the facts about the lysosome. So stay tuned. Very important. Vesicles is the small bubble within a cell, and the third type of organelle enclosed by lipid bilayer. So vesicles, they have just only one lipid bilayer enclosing them. So simply they are circular and they are enclosed by the single lipid bilayer. So vesicles can form naturally, for example, during endocytosis, or prepared artificially when they are called liposomes. So when the vesicles is enclosed by only one phospholipid bilayer, it is called a unicellular vesicles, or otherwise they are called multilamellar. So the vesicles, you can call them as unicellular vesicles or multilamellar. The phospholipid bilayer enclosing the vesicles is similar to the phospholipid bilayer enclosing the plasma membrane. So vesicles can fuse within the plasma lemma to leave their content outside the cell, but also they can fuse with other organelles within the cell. Generally, vesicles zinakuwa ni kama bubbles fulani within the cell, and they are used in different functions. So functions of the vesicles organize, collect cellular substance. Zinaweza collect vitu kwenye cell na kuvitransport kwenda sehemu nyingine. Vesicles act as chemical reaction chambers. Unaweza kukuta vitu fulani vinafungiwa kwenye cell, and there when they are enclosed in the vesicles, certain reaction takes place there. So they act as chambers of the chemical reaction. Vesicles are involved in the metabolism, transport, and buoyancy control of the enzyme storage. So zinahusika kwenye metabolism, transport pamoja na buoyancy control ya enzyme. Lakini here we have different types of vesicles. Now we have transport vesicles. These move molecules between locations inside the cell. For example, I told you that the proteins made in the rough endoplasmic reticulum, they are transported to the Golgi apparatus by the vesicles. And another type, we have secretory vesicles. These contain material that are to be exported from the cell. Example, synaptic vesicles in the synapse. They contain the neurotransmitter, and more especially is acetylcholine, which is more known, but there are other neurotransmitters like GABA and other which we shall discuss in the topic of coordination. So these, they contain neurotransmitter, and they release neurotransmitter out of the presynaptic membrane. And then you have gas vesicles used by bacteria and planktonic microorganisms, possibly to control vertical migration by the regulating the gas content and thereby buoyancy. So gas vesicles zenyewe zinakuwa zina gas, na kama unavyojua kwamba gesi ikiongezeka, gesi inallow kitu kifanye buoyancy, kielee. Kwa gesi kiongezeka kwenye ile gas vesicle, organism wanafanya buoyancy, anaelea, and then gesi kipungua, organism wana sink kwenda chini. So organisms, they have ability to regulate the content of the gas in the gas vesicle, and the organism have the ability to sink or to float. And then we have matrix vesicles, such as in paramecium. These are located within the extracellular space or matrix. Kwa hizi zinakuwa ziko kwenye matrix, hizi vesicles. Lakini pia we have vacuoli, ambazo vacuoli, these are vesicles which contain mostly water. And here we have different types of vacuoli. In animal cell, we have contractile vacuoli, which are found in certain protoctista, especially paramecium, amoeba, and they are used to take water from the cell and to avoid bursting due to osmotic pressure. Lakini pia in plant cell, we have large central vacuoli. In plant cell, it is also used in control osmotic pressure. Also, we have lysosome. Lysosome is also a type of vacuoli. Is also a type of vesicle. So lysosome, these are vesicles involved in the cellular digestion, destroying damaged organelle, that is autophagy, and death of the whole cell, that is it store hydrolytic enzymes for different processes, as we shall discuss soon. So now let's go deep into the lysosome. Lysosome, to study it as an independent organ of the cell. So the term lysosome comes from the two Greek words, lysis and soma. Lysis means split or break up, and soma means body. So lysosome, simply, it is involved in splitting or break up of the different bodies in the cell. That's one among its functions. So lysosome is the small cell organelle, spherical sac, bounded by a single membrane, which contain digestive or hydrolytic enzymes such as protease, lipase, nucleases, which break down proteins, nucleic acid, and lipids respectively. So protease break down proteins, lipase break down lipids, and the nucleus break down nucleic acids. So these organelles, their major function is to break out the different types of materials in the cell. However, they have other functions, as we shall discuss later. So lysosome is the cell organelle mainly deal with breakup of body materials or any aging organelle. So it can be body material or aging organelle. Lysosomes are found in eukaryotic cell except plant cell. They are also absent in prokaryotic cell. So they are found only in the eukaryotic cell, but not in plant cell. Plant cell, they lack lysosome. So in animal cell, they are abundant in animal cell which have phagocytic or engulfing foreign bodies and digestive activities. The enzymes contained within the lysosome are synthesized on larval endoplasmic reticulum and transported to the Golgi apparatus, where in the Golgi apparatus they are modified and packed in the Golgi vesicles, which are, in other name, they are called lysosomes. So the Golgi vesicles contain the processed enzymes, later body to form lysosome. So in plant cell, the large vacuole may act as a lysosome, although bodies similar to the lysosome of animal cell sometimes seen in the cytoplasm of the plant cell. So the vacuole is the synonym, ni mfanano wa vacuole kwenye plant cell. We call it synonym. So that's all about the lysosome. Lysosomes are made by the bodies, Golgi bodies or Golgi apparatus. The enzymes found in the lysosome are synthesized in the larval endoplasmic reticulum, as we discussed earlier, and transported in the Golgi apparatus in the cisternae. Golgi vesicles contain the enzymes body to form the primary lysosome. So here now we have different types of lysosome. Primary lysosome is the one which just buds off from the Golgi apparatus. It does not, or it is the lysosome which does not combine with anything. So maybe if the lysosome it combined with the other organelle for phagocytosis, now it is called the phagocytic lysosome, or it is called the secondary lysosome. So lysosome changes the name according to its content. So simply this diagram is not seen well, but it is simply the circular, then it contains the hydrolytic enzyme. So now let's go to look on the part of function. The first function, lysosomes are involved in the digestion of large molecule like food. So because the lysosome it contains the enzymes, so it is involved in the digestion of large molecule, and that's what we call the phagocytosis. Phagocytosis, by definition, it is the cell eating phagocytosis or endocytosis. Endocytosis is generally taking in materials, taking materials inside the cell, but phagocytosis is generally cell eating, so it is taking up the solid materials into the cell. That is what you call phagocytosis. So if your teacher asks you to maybe define phagocytosis is simply the process of taking in food materials into the cell by the lysosome. And another function is autophagy. So if you will look these functions in the biological science book, they are not written in this format, but I'm sure you understand them. Autophagy, lysosome is the one which is which is responsible for the digestion of the old organelles in the cell. So lysosome digests excess or worn out organelle, the process called autophagy. Here the lysosome is fused with the autophagic vacuole and bursts to release its concentrated enzyme which digests the content organelle itself. Or lysosome acts as a scavenger, ingesting and digesting the worn out cellular parts. So sometimes the organelle labda kama ni mitochondria inaweza combine na phagocytic vacuole, autophagic vacuole, then the lysosome ina combine na autophagic vacuole. Au sometimes lysosome yenyewe ina act kama scavenger. Scavenger ni kama ina meza hiyo mitochondria iliyoweka then ina digest. After the death of the cell, the lysosome are responsible for its complete breakdown, a process called autolysis. So in the functions of lysosome, autophagy and autolysis, there are two different functions. So autolysis is the self-digestion of the cell. That's why sometimes lysosome is called as the suicide bag. Suicide ni kama kujinyonga au kujitoa kafara, yani ni kujiua mwenyewe, kama vile ambavyo unasikia kwamba watu wanajiua wenyewe. That is called suicide. So it is called suicide bag because when it lies, ikipasuka lysosome ina release enzyme ambazo zinaiua seli yenyewe. Autolysis inaiua seli yenyewe. The enzyme causes the self-digestion of the cell. So that's all about the autolysis. Autophagy is the engulfing of the worn out organelle, but the autolysis is the self-digestion of the cell. Another function of the cell, here I have told you that lysosome is sometimes called a suicide bag. The third function, lysosome kills cells that are no longer wanted, such as those in the tail of the tadpole or in the web from the fingers 3 to 6 months of the old fetus. So lysosome, it is involved in the killing of the cell that are no longer wanted, killing of the cell. As I told you earlier that Golgi apparatus, it is involved in the apoptosis, but in the killing of the cells such as those in the tail of the tadpole, lysosome are also involved here in the process of killing the cell. So here, when the tadpole is changing into an adult frog, the lysosome in its tail cell called the breakdown of the structure, and the tail is absorbed into the frog's body. Also, when the cells are injured or die, the lysosome aid their disintegration. That's why lysosomes are frequently nicknamed that they are suicide bags or suicide sacks due to their autolysis. Sometimes it is called garbage disposal unit. The first function, lysosome also engulf and digest invading bacteria and viruses or any cellular debris. Debris ni kama uchafu. So any cellular debris, bacteria, virus ambazo zinaingia kwenye cell, lysosome ana engulf and digest. However, some of the bacteria and the viruses, they have ability, and not only some, most of the bacteria, they have ability to resist this digestion or engulfing. So that's what we call the pathogenesis of the bacteria. Pathogenic bacteria, they have ability to resist this, and that's why they have ability to infect human beings and cause diseases. Well, things are like that. Note, lysosomes are especially abundant in secretory cells and in phagocytic white blood cell. Lysosome play part in autophagy, autolysis, endocytosis, exocytosis. Lysosomes have many functions, also concerned with breakdown of structure or molecule. They are involved in the digestion process within the cell and also involved in the secretion of the digestive enzyme. In short, lysosome play part in the autophagy, autolysis, endolysis, and exocytosis. Note, single membrane of the lysosome prevent the breakage of lysosome content into the cell cytoplasm, because enzyme may digest even the cell, and that's why lysosome are sometimes known as suicide bags. So these functions of these lysosomes, if you go to another book, you will not see them in my previous lecture. You will see them in this format: autophag, autolysis. Autophag is the engulfing of the unwanted structure. Autolysis, self-digestion. Exocytosis, release of materials outside the cell. Then endocytosis. Taking in material. In endocytosis, we have two types: phagocytosis, that is cell eating, and pinocytosis, that is cell drinking. So phagocytosis is cell eating and pinocytosis is cell drinking. And here we have some of the diagrams which show the function of the lysosomes. But in my exercise book here, they cannot be seen well. You can see in the book of understanding biology, in the part of cytology, how that the lysosome, this diagram, you can see it in the understanding biology. I don't remember the page, but you can see how the lysosome, it engulfs, and here there are different stages from where it engulfs and it digests. And this diagram is found in the book of biological science. So I beg you to go and look at how that here we have endocytosis, then we have the lysosome. So we have vacuole here, and then the lysosome combined with this vacuole, endocytosis occur, and then the content remains, it's released out, that is exocytosis. And lysosome, it can be involved in the packing of the material, which can be released outside of the cell, or it can be involved in the autophag. So this is the autophag, this is cell digestion, exocytosis, this is exocytosis also, this is autophag, and it simply summarizes the function of the vacuole. So as I told you earlier that we have different types of vacuole: primary, secondary, and etc. etc. So I beg you to go through the biological science book. Here my notes, I have not modified them. When I will modify them, I will update this lecture, but according to the need of my students. Now you may use this to get not a literal, because most of teachers, they don't teach these parts in school. So don't just listen and say this teacher is teaching shallow, and then when you go to school, you will reach and you will meet different teachers which are teaching in different style from my style. So sometimes it can make you difficult to understand, because they are skipping most of the things. Okay, for this lecture, thank you. Next lecture, we shall be discussing about the microbodies, that are peroxisomes, and don't miss it. Very important. It's very, very enjoyable to understand well the cell. If you understand well cytology, it will be easier for you to study other topics. And when I will be going into other topics, I will just tell you that as we discussed in cytology. So if you didn't understand cytology, it will be very difficult for you to understand it. And most of the time, we say that I'm not teaching well, but the problem is you and your understanding. Thank you, everybody. Have nice studies, and subscribe the channel for the more updates. Meet you next lecture.